Thanks very much. And thank you very much for the organizers for bringing us together here. It's really a wonderful opportunity, especially for someone like me who is a relative newcomer to this field of biomolecular condensates. And so really learning at this interface of experiment, simulation, and theory is really great. And it's something that I've appreciated a lot over the last 15 years or so, that we've worked on intrinsically disordered proteins and their behavior. And especially we've been interested in the behavior of highly charged intrinsically disordered proteins. And so one of our most important examples that we've worked with so far is this pair of proteins. This one is histone H1, which is one of the linker histones that compacts the nucleosomes and on a large scale leads to chromatin condensation. And it's a highly positively charged protein full of lysine residues and has a net charge of plus 53. And its binding partner, prothymosine alpha, one of its binding partners, prothymosine alpha, a highly negatively charged IDP full of acidic residues. And so these are two full-length human proteins that are known to interact in the nucleus. And they've been known to interact for 25 years or so. But what wasn't clear at the time is what would happen when these two proteins bind to each other. And so in a combination of single molecule FRET experiments with CD spectroscopy and NMR spectroscopy done in Birgit Grachigon's lab in Copenhagen, we found that the two proteins bind to each other with very high affinity. So we're talking about picomolar to nanomolar affinities in the physiological strength range, but that they don't form any structure when they bind to each other. So this is illustrated here with these coarse-grained simulations done by Robert Best that are in good agreement with the experimental data. And so in the middle here, you see the globular domain, which histone H1 has, which is important for positioning it on top of the nucleosome. But you can actually delete this folded domain, and it has very little influence on the affinity. So the affinity is really dominated by the charged disordered regions of these two proteins. And so we also know that this interaction is functionally important. So prothymosine alpha acts as a linker histone chaperone. So what that means in practice is that it accelerates dissociation of histone H1 from the nucleosome by 2 orders of magnitude. The spontaneous dissociation of H1 would be too slow. And so from a biological point of view, what was remarkable about this type of interaction is that you can have a very high affinity interaction without the formation of structure. So if you come from a polymer physics background, it may not be so surprising to you that two oppositely charged polyelectrolytes bind to each other with high affinity. But what might still be remarkable from a polymer physics perspective is that you could think of this as sort of the most elementary complex coacervate of just consisting of two molecules. And as far as I know, this is something where polymer physics can benefit from biology, because I don't know of an example where the interaction between two molecules of synthetic polyelectrolytes could have been studied. But since we do single molecule experiments and we have all the labeling methods available and the like for proteins, we can study this kind of biomolecular complex coacervate. And so if you then go to higher concentrations, then you go away from this simple one-to-one complex coacervate and you form classic complex coacervates. You phase separate, and you can characterize these complex coacervates with established methods. You can measure phase diagrams here as a function of salt concentration. One of the interesting pieces of information from this is, we get here from the right binodal, the protein concentration in these droplets is very high, in hundreds of milligrams per milliliter. And we can also use standard rheological methods, like nanorheology here, bead tracking, to get information about the viscosity of these droplets. And here, in this case, we get 300 millipascal seconds, so 300 times the viscosity of water. And so this is a number that I would like you to remember, because we need it again in a little bit. But the key step, or the key goal, was really to try and get an understanding of the conformational dynamic properties of these IDPs inside these droplets. That's sort of one of the key motivations for our experiments. And the key step towards doing this was done by Nicola Galvanetto, a fabulous physics postdoc in the lab. And the idea was to do single molecule FRET experiments inside these droplets. And so the idea is you phase separate and you form the droplets with unlabeled protein, and then you add a tiny little bit of protein labeled with a donor and an acceptor dye, so we can do single molecule FRET on these molecules. And I guess you're all familiar sort of with the fundamental idea of FRET experiments, right? Where you can get distances between these molecules labeled. And so then you can focus your laser beam into the solution and either measure here in the dilute phase. And so then you get the typical signal that you get from molecules freely diffusing in solution. And so every burst of fluorescence that you see here corresponds to an individual molecule diffusing through the focus, which takes on the order of a millisecond. That's sort of the typical translational diffusion time through the confocal volume. But that's relatively easy to do because the concentration of protein is very low. It's, for technical reasons, quite a bit harder to do this inside droplets, but conceptually it's very simple. You just focus your laser beam into a droplet, and then you have your confocal observation volume, and you look at the molecules inside the droplet. And you can already see from the raw signal trace here, you see the qualitative difference. Now these bursts of fluorescence are much longer than in the dilute phase, which is what you would expect because the viscosity is much higher, right? So they just diffuse much more slowly. And so based on these data, we can then extract information about the behavior of the molecules. And the first thing we can do is we can measure these transfer efficiencies with nothing but the probability of energy transfer between donor and acceptor. We can relate that to an average distance between the two dyes attached to our molecules. And so we can compare prothymosin alpha free in solution, where we observe a transfer efficiency, if you look at this peak here, that is relatively low, which is what you would expect because it's a highly charged protein, and so there's charge repulsion. When you bind it in the one-to-one complex to the positively charged histone H1, then of course prothymosin alpha becomes more compact, the transfer efficiency increases, and then if you measure prothymosin alpha inside the dense phase formed from prothymosin alpha and H1 in high concentration, you get a transfer efficiency sort of right in the middle between these two cases, and then we can extract information about distances, and from this, for instance, infer an average root mean squared end-to-end distance of the chain, right, which is on the order of 9 nanometers in this case. The second piece of information we can get is about the dynamics, right? So what people use FRET very often for is to get an average distance. But it turns out you can not only get information about an average distance, you can also measure distance fluctuations. Because if the two dyes fluctuate in distance relative to each other, the transfer efficiency also fluctuates, and correspondingly the fluorescence emission. And you can measure these fluctuations in fluorescence emission by correlation spectroscopy. And by using some technical tricks, we can push these measurements into the nanosecond range, and then we call this nanosecond fluorescence correlation spectroscopy. And these are examples of such measurements where it is this component here, so this drop on the nanosecond timescale, that reports on these distance fluctuations. And so if you do this for prothymosin alpha alone, you get a relaxation time on the order of 10 to 20 nanoseconds. So very rapid, but this is sort of what you would expect if you take a simple Rouse or Zim model with realistic parameters. That's the kind of dynamics you expect for a single chain. If you now bind prothymosin alpha to H1, the time scale increases by about an order of magnitude. And the reason is that the intrinsic dynamics of H1 are much slower. And so essentially prothymosin is kind of slaved by the binding to H1. And now the most interesting question was, what happens to the dynamics in the dense phase? And now remember that we had a viscosity in the droplet of 300 millipascal seconds, 300 times the viscosity of water. So the question was, would the dynamics also be slowed down by a factor of 300? And then the surprise was that this is not the case. It only slows down by about a factor of 3 when you go into the dense phase. And so this was the first question that we had to answer. How can it be that we have a bulk viscosity that's 300 times higher than a dilute solution, but the chain dynamics are only about 3 times slower? And so there are two answers that we know about. The first answer is that, of course, we're talking about a complex network fluid. So the viscosity is no longer a length scale independent quantity. And so if we do the following experiment, that we measure the viscosity, or the effective viscosity, we call it here, using the Stokes-Einstein relation, as a function of the hydrodynamic radius of the probe particle. Then if you use sufficiently large particles, you measure the bulk viscosity, or what's called here the macroscopic viscosity, right? That's sort of the classical rheology experiments. But then if you go down in size, you see this drop in the observed viscosity, and for the smallest size scales that we're able to measure experimentally, we're already pretty close to the solvent viscosity, right? So it's very clear viscosity depends on length scale. And the quantity that is used to characterize this kind of transition from the microscopic to the macroscopic viscosity is the correlation length. And we can estimate the correlation length based on this little equation. So we have the radius of gyration of our molecules, which we know from our single molecule FRED experiment. We have an idea about the dimensions, so we can estimate the RG. And by the ratio of the protein concentrations that we have in our droplets relative to the overlap concentration, which we can also estimate for our molecules, for our dense phase. And then we get a correlation length that's in the range of 3 to 4 nanometers, so the purple band here. And so we can now use these three quantities, the solvent viscosity, the bulk viscosity, and the correlation length, and plug it into a theory that was developed by Remco Tournier 20 years ago. And this is the result. So it sort of describes this behavior very well. This is a theory built on depletion interactions. And it sort of really shows, I think, very naturally how we go from this local viscosity, which depends very much on which length scale you're exploring, to the bulk viscosity, where a particle has to move through the entire network of the molecules. And so if we now ask, where does prothymosine alpha that we measured sit on this length scale? This is where we are. So we're right in the range of the size of our molecule. And you can see here that the effective viscosity that we measure on the length scale of prothymosine alpha does explain some discrepancy to the bulk viscosity, but it doesn't go all the way, right? So maybe it explains 1 order of magnitude, but it doesn't explain a factor of 300. So there must be something else going on. And so to see whether we can identify any effects on the molecular level, Miloš Ivanović, another fabulous postdoc in the lab, in this case with a background in MD simulations, he decided to do large-scale MD simulations in close collaboration with Robert Best. And so this is the simulation setup. We have a large simulation box containing almost 200 of our proteins. All of the solvent molecules is explicit solvent because we want absolute timescales in this case, right? This is the key information that we're after. All of the water molecules, all of the ions, altogether 4 million atoms, using Robert Best's force field for IDPs that we already know from other simulations that do a pretty good job. And so in 6 months on the Swiss supercomputer, he was able to get 6 microseconds of simulation time. Of course, we only dared to do this because we knew from the experiment that the dynamics are on the hundreds of nanosecond time scale. So we had some hope that we can get something reasonably converged that we can compare to, right? And so now we can check whether these simulations are any good, and we can compare several quantities like the protein concentration in the dense phase, the translational diffusion coefficient of prothymosine, the transfer efficiencies, and let me focus here on the chain reconfiguration time. So these chain dynamics that we measured experimentally, and here are the 380 nanoseconds that I showed you, and the simulations are really in the right range. And so that gives us some confidence that we can learn something from the simulations. And so then let me show you 2 results that Miloš extracted from the simulations. The first one concerns the interactions that these chains make in the dense phase. And so what's plotted here is the average number of contacts that every residue in prothymosine alpha makes with the other chains in the dense phase. And what's plotted here is the profile in gray here for the 1-to-1 dimer and in purple for the dense phase. And you can see that the profiles are very similar, which tells you that for the individual protein, it doesn't make such a big difference whether it's bound to one H1 or to many H1s, right? On average, every prothymosine chain interacts with 6 H1 molecules in the dense phase. The second piece of information that was interesting and quite surprising is that we now look at the lifetime of contacts between side chains. And here again, we have a comparison in gray of the one-to-one complex and in purple of the interactions in the dense phase. And you can see that, again, they are very similar in the two situations. And most importantly, they're in the low nanosecond range. So there are no persistent salt bridges, but these salt bridges are very short-lived. And so the question is, why is that? And for that, we need to first realize that the concentration of charged residues in the dense phase is extremely high. We have an effective concentration of about 1 molar of charged side chains in the dense phase. And so if we now look at the simulations on this timescale of a few nanoseconds where these kinds of processes seem to occur, we see that something that happens quite frequently are processes like this, where you have here a salt bridge between a positively charged residue and a negatively charged residue. And then here comes another positively charged residue. They form kind of a ternary interaction, which is easily possible because of the long-range nature of the electrostatic interactions. And then the original side chain here gets kicked out. And so we have sort of a rapid exchange, or as Paul called it, dynamic shuffling of these interactions in these complex coacervates. And that seems to contribute to these dynamics of the chain that are retained even though the bulk viscosity is very high. And I think that especially in situations like the nucleus that is packed with polyelectrolytes, these kinds of dynamics might be very important to keep the nucleus as fluid as it is and as functional as it is. And so to see whether this is something that is more general, we decided to go beyond the two proteins that I talked about so far and expand the range a little bit to other sequences. And so there is another type of molecules, positively charged proteins, that are very important for DNA packaging, and these are the protamines. And so these protamines are, for instance, important in packaging DNA in sperm head, where the DNA is packaged much more tightly than in a regular nucleus. And if you look at the sequence of these protamines, you will see that they're packed with arginine residues. They're essentially polyarginine. And so we wanted to see how much difference does this make, the lysine versus the arginine. And so for comparison, we also included polylysine and polyarginine to see how much difference we can get. And so we have the following 4 pairs that we're looking at. We have our negatively charged prothymosin alpha, and we're now combining it with our 4 positively charged interaction partners. And all of these guys form beautiful droplets. All of the experiments I tell you about are done where the proteins are mixed at charge balance, which is where phase separation tends to be most pronounced. And I should also mention that we observe no aging in these systems, right? Which is different from many, especially hydrophobically driven condensates, where you do see aging and other processes. This doesn't seem to be the case here, which simplifies the experiments greatly, of course. And so let's look at the phase diagrams of these different guys. So this, just as a reminder, that was the phase diagram for H1 and prothymosin alpha, so that's our reference point. And so if we compare this to the phase diagrams for the other guys, it's pretty obvious that especially if you include arginine-rich sequences, and most pronounced for arginine 50, you get phase separation up to salt concentrations of 1 molar, whereas for H1 and prothymosin alpha, we've not been able to get phase separation even at 200 millimolar salt. So very clearly, arginine content and increase in charge density increases the driving force for complex coacervation. And so what about the dynamics? Right? So this is really what we're most interested in. How do the dynamics change? And if I say dynamics, I mean dynamics not only on the mesoscopic scale. This is what most commonly is measured in FRAP experiments and droplet fusion, but also on the molecular scale. But let's start with the mesoscopic scale. So if we do microbiology, or Nicola does microbiology, and now we're plotting, what he's plotting here is the viscosity, the bulk viscosity, as a function of the salt concentration. And so for reference, H1 and prothymosin alpha, the example I showed you before with the 300 millipascal seconds is down here. And now if you decrease salt concentration, the viscosity goes up, which is not unintuitive, but a much more pronounced effect is what you observe if you go to arginine-rich sequences where the viscosity increases by more than an order of magnitude. Next level down in size scale would be to measure translational diffusion of prothymosin alpha. This is something that we can measure very well using classical fluorescence correlation spectroscopy, where you just measure translational diffusion through the confocal volume. And so plotted here in terms of the diffusion time through the confocal volume, again, H1 prothymosin alpha down here on the time scale of a few milliseconds, essentially. And if you go to the arginine-rich sequences, you increase the diffusion time, again, by more than an order of magnitude. And the last length scale, at the molecular scale, now we're measuring the chain dynamics of our system, of our little prothymosin inside the droplet. And we reported here in terms of the chain reconfiguration time, our H1 prothymosin alpha with its 400 nanoseconds will be down here. And you can see again, especially for the arginine-rich sequences, low salt, we have reconfiguration times that are now in the 10 microsecond range, so much, much slower. So across all these scales, we see a pronounced effect of especially the sequence and also the salt concentration. So the dynamics across all of these scales are strongly affected. And if we plot them in direct comparison, our three quantities, you can see that they're highly correlated in all these cases, which already indicates that there may be a mechanistic link between them. And so the first question was now, what happens at the true molecular scale? So Milos again ran some simulations now with protamine, and you can probably see it a little better on the screen here. That's a little more brilliant. And what's very obvious is the slower dynamics of prothymosin alpha in the combination with protamine. And it's approximately right change in time scale, but of course, then it's very difficult to really converge long scale motion. But sort of getting the local interactions converged, that still is something that we can do. So we can look at the interactions in the similar way that I showed you before. And so we can now compare again these sort of number of contacts that prothymosin alpha makes. The blue line here is what I showed you before in the interactions with H1 in the dense phase. And you can see that decreasing the salt concentration changes things a little bit, but a much more pronounced effect is if you now go to protamines, the arginine-rich sequence, where you can see that the average number of contacts that every residue makes increases by almost a factor of 2. So many more interactions in the dense phase. Then the question is this dynamic shuffling still going on in these sequences? And so this is what I showed you before with lysine-rich sequences, right? So here we have this dynamic shuffling very clearly, but also with arginine it happens, even though the arginine can make these sort of divalent interactions, if you like, but it still happens. You still have a rapid exchange, but it's clearly slower. And so if we compare the contact lifetimes between the interactions with H1 in blue and arginine in purple, you can clearly see that the lifetimes are longer. And if we compare them quantitatively, it's almost an order of magnitude in the average contact lifetime. Now, this has an important implication, which is that we have a separation of timescales between the making and breaking of these individual interactions and the chain dynamics. Remember that the chain dynamics are here on the hundreds of nanoseconds or here on the microsecond timescale even. So that means that in practice, during one chain reconfiguration time, in this case, we're making tens of thousands of interactions. Right? And so in this case, it seems that we're going from sort of the sticky Rouse model in the spirit of Semenoff and Rubinstein, we're going towards the classic Rouse model because this behavior is very similar to an effective increase in friction. Right? And that's sort of the basic idea of the Rouse model. So let's see how well the simple Rouse model does in this case. And so here we now have the viscosity measured by microrheology, so the bulk viscosity as a function of the diffusion coefficient of prothymosin alpha in our droplets for our 4 different combinations. That's the data. Now, this is what simple Rouse theory would predict, and though that's essentially textbook polymer dynamics. So many of you would probably consider this kindergarten polymer physics, but it's still not so easy for us experimentalists to distill down the details of what we need to use. And so the idea here is that the viscosity as a function of the diffusion coefficient depends, of course, on the solvent viscosity. Then we have Kt over 36, and here we have the protein concentrations. We expect the viscosity to change linearly with protein concentration. We have the mean squared end-to-end distance of our chains. And we know the protein concentrations that we measured in the dense phase. We know the mean squared end-to-end distance because that we estimated from our Fred experiments. So we can just plug in the numbers and compare with the data. And you can see, well, it's not so bad. It's sort of within an order of magnitude of the experimental data. It gives you the right slope. So I think that's all pretty good. But we're clearly off. So the question is, what could be going on? And of course, if we look at our concentrations, we have to realize that we are, in most cases, above the overlap concentration. Right? So this is no longer a simple solution, but we probably have a weak entanglement contribution of some sort. And so that the simplest way of including entanglement in this simple Rouse model is with this correction term here that depends, again, on the dimensions of our chains, and then this quantity A here, which is what Dejean and Edwards originally introduced as the tube diameter in their reptation models. Now here, of course, there is no true reptation because our chains are very short, but still, there is something, I mean, some people refer to this as the entanglement spacing, which maybe is a better terminology in this context. And so this is, we could consider this as an adjustable parameter, but we have an idea of what it should be approximately, right? Because we expect this length scale to be similar to the correlation length of the system, right? And so if we treat it as an adjustable parameter and try to fit the data by varying A, this is what we get, and we get a value of A that's about 4 nanometers. So not unreasonable, yeah? So I'm not talking about deep entanglement, but I think some of these contributions may be important in this case. And so now we can also try to compare to other systems because there are a few more systems for which we found data where both the viscosity and the diffusion coefficient have been measured. So here's LAF1, FUS LCD, and A1 LCD here with Tanya's work. And you can see they're pretty close, right? So I think it's probably a reasonable first order approximation to what's going on in these systems. So this would be one relation. Let me show you one other example. And now we're comparing here the viscosity, bulk viscosity versus the reconfiguration time. So really the molecular scale dynamics. And if we use the Rouse model again, this is the relation that you find. And now we know A should be 4 nanometers, so no adjustable parameters. And this is what you get, right? So not so bad. And here we could not find published data on any biomolecular system, but we found these data from the Schlenov lab on 2 synthetic oppositely charged polyelectrolytes, so complex coacervates of synthetic molecules. And so they inferred in a way very similar to what we heard from Ellie yesterday, the chain dynamics from oscillatory rheology. And they find millisecond chain dynamics, right? So it's orders of magnitude slower than what we see in our experiments. But if we just extend our line here, you can see they sit on the same line, which could be a coincidence, but maybe not, right? Maybe there is really more to it. And maybe the simple Rouse theory does provide sort of a simple universal link, at least to a first order approximation. And so with that, I hope I was able to show you that the combination of experiment, simulation, and theory can tell us something about mechanisms in these systems. And so I'd like to summarize with this little overview. What was plotted here is the prothymosine concentration on this axis, the H1 concentration of this axis, and we started out with this 1-to-1 complex. Right? And so this is the species that's dominant in the dilute phase. That's our elementary complex coacervate, if you like. And we can probe it here because we're doing single molecule experiments. And we can go to sufficiently low concentrations that we can even measure the association, dissociation, equilibrium between these guys. If you go to a situation where you have an excess of one of the 2 proteins, then you can imagine you can form these ternary complexes where you have 2 of one and 1 of the other. And if you go to very high concentrations up here in this corner, then you get complex coacervation between the two proteins. And what I showed you was that we have these rapid chain dynamics, even though we have a large bulk viscosity. And there are at least two contributions that are important here. One is that we have length scale dependent effective viscosity because we're dealing with a network fluid. And the second is, there's something that we learned from the simulations, that we have these very rapid exchange between the side chains in the dense phase. And I also showed you that this behavior depends a lot on the sequence composition. Arginine especially slows down the dynamics, but that we can use very simple polymer theoretical concepts to explain these dynamics across the scales. And so the missing link that I think we will, or I hope we will be able to close soon, is to relate this directly to the simulations. And these are ideas that, for instance, Rohit has also been working on. And so here what you see is the correlation between the contact lifetimes in the simulations to the chain reconfiguration time. And I think this correlation sort of makes us optimistic that eventually we should be able to take simulations and predict the mesoscopic properties of these systems. And so with this, I would like to thank the fabulous team that contributed to this work. Nicola spearheaded the experimental part and a lot of the theory in this work. Miloš did the simulations on the complex coacervates. There was some help from Simone, Alessandro. Alessandro Madeleine actually started this work on H1 and prothymazine almost 10 years ago now. And a lot of other people helped here, Ritra, Andrea, two Andreas. Peter did the work on the nucleosomes. Davide did simulations. Daniel is a key person for the technical side on the experiments. Robert Best either did simulations like the ones on the one-to-one complex or was a close advisor for Miloš for doing all of the other simulations. And I should also mention Birde for her collaboration with the NMR experiments. Thank you very much.
Thank you. Are there questions? Yes, many questions. Okay. Just over here.
You made this distinction between Rouse relaxation and sticky Rouse relaxation, but then you didn't come back to that. Did you see, were you able to make that distinction in any way? And particularly, did the arginines and lysines, was there any difference there?
So in both cases, we are in a regime where sticky Rouse does not really apply. Right? So sticky Rouse applies in the situation, where do I have it here? Sticky Rouse applies in the situation where the lifetime of the stickers is much longer than the relaxation of the chain between them, like in the example that Ellie showed yesterday. But here, it's the opposite, right? So the contact lifetimes are orders of magnitude shorter than the relaxation of the chains.
Great, okay.
And so that's why we think we're not in a regime. And I discussed this in detail with Michael Rubinstein also, and he said, clearly, it doesn't apply in your case.
Fantastic.
He was very decided on this.
There were questions over here.
Thank you for the very clear talk. Could you just say a few more words about the salt concentration? Within the condensate, are the ions that come from the two charged proteins still at a much lower concentration than the salt concentration if I look at 150 millimolar and above, or are they comparable? Because there are all kinds of funny things that happen at high salt concentrations in simpler systems. And I'm wondering, what is the effective ion concentration within the condensate?
Yeah, so we've started experiments to measure the ion concentrations, but at this point, the best I can offer is what we get from the simulations. And so maybe if you look here at the case of H1 and prothymazine alpha at 128 millimolar KCl, which was sort of the conditions of the first part that I talked about, then you can see here the number density of ions outside of the condensates, and here that would be inside of the condensates. And so the number density is slightly lower in the condensates in this case.
Because of the counter ion release.
Yes, so there is some counter ion. I mean,
Plus minus, you could release counter ion.
most of the counter ion release already happens in the dimer formation. Yeah. But then there is still some net charge because they are slightly asymmetric. Right. And so there is a little bit of additional ion release upon phase separation of the dimers into the dense phase. Yeah?
More questions? I'll go back there and then come back to you.
It's really beautiful, as always, Ben. I had a question about, so the chain reconfiguration time, sort of how you calculate it. And what I'm thinking about is that while the local dynamics seem very rapid, it seems like the larger intrachain entanglement maybe has a much higher correlation time. And I'm sort of wondering if that affects the experiments, or if there's enough flexibility within a given entanglement that it matches the correlation times of the FRET probes, or so if you're basically how are you computing interchain correlation time? Is it just monomer to monomer, or do you have a longer interchain measure?
So in this case, or in the case of H1 and prothymosine alpha, we can do the very simple thing of just calculating the distance correlation function, right? And that is essentially equivalent to what we measure in the experiment. And the convergence is good enough, especially also because we have 200 chains here, right? Or 100 of one case. And so if we use all of these, then we can get a pretty good number for the chain reconfiguration. Yes, yeah, yes. And you can then average this over all molecules in the slab, and that gives us a reasonable convergence here. And in that case, it's pretty hopeless to do this. So in that case, the trick that Rob suggested to use is just treat this as diffusion and a potential. So we just approximate this behavior as diffusion and a potential, and then see which effective diffusion coefficient we have to use to be in agreement with the simulations, so that the error bar is significantly larger, but it's in a reasonable range, I would say.
So question about the reconfiguration time scheme. So you have about a 200-fold increase in this reconfiguration time of H1 in the presence of protocysteine versus protamine in the presence, which clearly indicates a much stronger interaction of arginine-mediated salt bridges.
Yeah.
But you also measured the H1 on its own. I think it had 14 nanoseconds.
Yeah, the prothymosine alpha alone, exactly.
Right.
We also measured this alone.
Right.
And there you have a very rapid configuration.
Right. So what would you expect a similar measurement would give you for just protamine on its own? Would it be something similar, or it's also can be very different?
So the question is essentially, if we take prothymosine alpha and we just put it into a high viscosity solution.
No. Do the same for protamine. The
Also
arginine-rich.
measure protamine.
Right.
Yeah.
For arginine-rich single chain, would that reconfiguration time on its own be different from a lysine-rich sequence?
So in this case, we can only tell from the simulations, because we've not yet been able to do the experiment on the arginine peptide, because it's not so easy to make it and label it. But from the simulations, the reconfiguration times are, well, actually, I'm not sure I know the numbers. I seem to remember that the reconfiguration times were very similar for the protamines as they are for the prothymosine. But I would have to check the numbers. We can look at the numbers from the simulations.
Sorry, I'm going to try to go in order. So.
Yeah, Ben, lovely talk. I know this might be a hard experiment, but have you thought about trying to directly measure the correlation length with scattering?
Yes, we're actually in the process of doing that. So a postdoc in the lab, she's currently doing experiments in collaboration with Eric Dufresne, where we're trying to do scattering experiments too. And so the preliminary results look like the correlation length they get from scattering is similar to what we infer from our length scale dependent diffusion. But the final data are not yet in, so I don't want to stick my head out too far.
Lovely talk. So the parameter A that you find, so this should also be system dependent, right?
Yes, that's right.
And so I'm quite surprised that when you took those other systems and put them on the same plot, you got remarkably good agreement. So well, if you could just comment, do you think that there's some deeper reason that things seem to
so
match up
the
so
reason
well?
we did this is just that we could put everything into one plot. So what you'd have to do, strictly speaking, you'd have to plot something like a reduced viscosity, where here we divide by the protein concentration and the chain dimensions for every system individually. Right? And then for every system, you would get a separate fit, right, and the separate value of A. And these are the tube diameters, and they're somewhere between 4 and 5 nanometers. So the variation is not large, but you're absolutely right. It's slightly different for every system, yes. So we just sort of, given the relatively large error bar, we just decided to just put them into one plot.
Any more questions? Okay, going once, going twice. We are actually on time, so the next speaker can come up and set up. Thank you. Let's thank Ben again.